So first of all, I wish to thank Norberto for inviting me here. It has been a really great meeting. And then let's get in. I'm going to talk about gene therapy for epilepsy. You all know that epilepsy has a number of unmet therapeutic needs. We clearly need treatments for drug-resistant seizures. We need treatments with increased tolerability. And of course, we need treatments to prevent or ameliorate epileptogenesis or comorbidities. What I will try to do today is to, I'm not sure if I convince you that gene therapy may represent an effective alternative to standard pharmacological approaches, but I will try to critically summarize all the data that have been obtained so far using this approach. So this is an outline of my presentation. I will start describing the possible gene therapy intervention in the epilepsy spectrum disorders. I will then get into the choice of the vectors and the choice of the route of delivery. I will briefly mention the models and the way they've been used for gene therapy studies. And then, as Ashok mentioned, I will speak more in detail about possible anti-epileptogenic effects. This is because data come mainly from my laboratory, but I will also speak about anti-seizure effects that have been obtained using gene therapy approaches, and then I will try to propose some ideas for future developments. So, first of all, possible gene therapy interventions. When you talk about gene therapy, of course, one idea that some may have is that since a large percentage of epilepsies are genetic in nature, then gene therapy should be particularly useful for the genetic forms of epilepsy, which is actually not the case. There are two fundamental problems with genetic epilepsy. The first one is that they are rarely caused by a single mutant gene. Genetic epilepsies require the inheritance of two or more predisposition susceptibility genes. The second problem is that many of the genetic epilepsies have a widespread pathology that affects large portion of the brain. And so that poses a problem in terms of widespread delivery of a gene therapy vector. In the future, it could be possible to approach even this kind of problems because there could be, actually there are under development some gene therapy approaches that may allow the vectors to enter the brain in a diffuse manner after peripheral administration. And actually the most promising of this is called transcytosis. I'm sorry, I did it wrong. What is transcytosis? Essentially, transcytosis is a mechanism that employs a pathway used by circulating endogenous models molecules like transferrin or insulin to enter the brain. What do these molecules do? They bind to their specific receptors at the vascular site of the endothelium, and the complex of the receptor and its ligand is engulfed into a vesicle. The vesicle is transported across the blood-brain barrier, and the ligand is released intact in the brain site. So it is possible to develop this kind of approach also for vectors, and I'm talking specifically about viral vectors, because the idea here is to engineer the surface of the vector such that you have a conjugation with a ligand for one of these receptors. Could be a single chain antibody or a peptidic mimetic. So this way, the vector should bind the receptor, undergo transcytosis, and be released intact in the brain. This possibility has been tested using adeno-associated vectors with promising results. These vectors have been demonstrated to undergo transcytosis, at least in the rodent blood-brain barrier. But these vectors have not been tested in models of genetic epilepsy so far. So this is a possibility, but in general, there's much work to do that remains to be done for the genetic treatment of genetic epilepsies. And more feasible is the approach to local, focal lesional epilepsies. And this presents a number of advantages and opportunities. First of all, pathology is focal, and therefore stereotaxic inoculation is possible. Often, it is possible to identify a cause for the diseases, a trauma, for example, or a stroke, or other. And therefore identify patients that are at risk for development of epilepsy, and as you know, we have models of this situation. So in general, there's opportunity for intervention at different levels, preventive, so anti-epileptogenic, symptomatic, anti-seizure, or disease-modifying that would include also treatment of comorbidities, but as I said, I will not really talk about that today. The second point is the choice of the vector and of the route of delivery. There is no doubt that vectors for gene therapy are today all based on viruses. There is nothing that can match the highly evolved biology of viruses and their capacity to infect cells and express their genes in the cells. So it is based on the elimination or attenuation of the capacity of viruses to reproduce in the cells, and to the introduction of therapeutic genes in the genome that one can approach gene therapy. As I think most of all of you know, there are multiple virus backbones that have been used to develop vectors, and each of these have advantages and disadvantages. In general, the viruses that have been used for applications of gene therapy to epilepsy are either adeno-associated viruses, lentiviruses, and herpes viruses. Just to mention a little bit of the advantages and disadvantages, adeno-associated viruses are non-pathogenic and therefore are safer than the others, and ensure a long-lasting expression of the transgene. But the downside is that they are very small viruses. Their size is about 5 kilobases, and therefore there is room only for insertion of a single therapeutic gene, which is something that applies also to lentiviruses that also have the advantage of a long-lasting expression. But on top of that, lentiviruses also have another problem, that they insert their DNA in the host cell genome, exposing to the risk of insertional mutagenesis. Herpes is instead not inserting its DNA in the cell genome, but has the important advantage, I believe, of being much larger than any other viruses. The genome of herpes is composed largely by non-essential genes that can be replaced theoretically with therapeutic genes, and it is about 30 times larger than the one of adeno-associated viruses. And so that herpes is the one vector that offers the possibility of creating composition of therapeutic genes and inserting them in the very same viral particle, therefore expressing the transgenes all together in all infected cells. As for the route of delivery, it is clear that the stereotactic injection in the epileptogenic region is by far the one that has been used the most. This is an approach that ensures a high level of transgene expression, a relatively limited immune response, even if the breakdown of the blood-brain barrier may induce the penetration of lymphocytes. And it is also possible, based on the biological characteristics of the viruses, to modulate the spread from the site of injection. For example, various adeno-associated serotypes have been developed that can spread more or less from the site of injection. And herpes has the capacity of entering the nerve terminals of neurons, thereby being transported retrogradally to anatomically connected regions. For example, if you inject herpes in one hippocampus, you can have expression of the transgene in the contralateral hippocampus because of transport through commissural fibers. Of course, a problem with this kind of approach is that it requires stereotactic surgery. So there is research that is ongoing in an attempt to develop alternative approaches that avoid this invasive one. And one approach that has been tested is the one of the intranasal injection of vectors. These are generally herpes vectors that can be transported from the mucosa to the bulb and to other regions in the brain. Of course, in this case, the problem is that you don't get high levels of expression of the transgenes, and two, that you do not have selective expression of your gene of interest in the area of interest. Another approach that is very promising has been developed, unfortunately, 3 years ago, and I have not seen any further advancement after that paper by Steve Gray and McCall at Chapel Hill. And what they found, they selected a different kind of chimeric adeno-associated viruses and tested them for their capacity to penetrate the broken blood-brain barrier in an epilepsy model, namely kainate. And they found one serotype that was capable of doing this. Of course, this would be very interesting because it would mean a possibility to induce overexpression of a gene of interest selectively in the area where you have breakdown of the blood-brain barrier that could be the epileptogenic area. Just a couple of words on the models. The models that have been used the most are kindling and status epilepticus induced epileptogenesis. As you all know, these models allow evaluation of anti-seizure effects and disease-modifying effects. It is often said that they can also evaluate anti-epileptogenesis, but in the case of adeno-associated vectors, this is not really possible, or best, it is not unequivocal, because it takes several weeks for adeno-associated vectors to express the transgenes, and therefore in the studies where when
these have been used, they were injected weeks before induction of status. And therefore, it's probably safer to say that those effects were anti-seizures, even if they could be anti-epileptogenic. So, moving to the results, gene therapy approaches have been used to attempt to obtain anti-epileptogenic effect, and it has been done mostly in my lab in a series of papers in which we tested a combination of two neurotrophic factors, the fibroblast growth factor 2 and BDNF. As you know, we have been discussing about that in these days. Epileptogenesis is associated with a number of adaptive changes in the tissue, which include cell death, increased neurogenesis with a more or less large component of aberrant neurogenesis, and neuroinflammation, and many other. The concept here was, what we reasoned was that if we produce some kind of neuroprotection and induce a better level of neurogenesis, we may obtain an anti-epileptogenic effect. And the idea, to make the story short, because was a lot of proof of concept before this, was to use a combination of FGF2 and BDNF. Both these neurotrophic factors have, in fact, neuroprotective effects. FGF2 is also the single most potent proliferation agent for neural stem cells in the hippocampus, whereas BDNF is considered one of the best characterized agents capable of inducing neuronal differentiation of precursors. And by the way, there was some evidence when we started that a combination of these two factors could be useful experimentally, and some of these came from the lab of Ashok Shetty. So what we did was to use herpes. Again, we needed to use herpes because we wanted to express two transgenes, and this is the genome of herpes, and this is the way it is regulated. As you see, herpes induces expression of immediate early genes that are transcription factors that regulate expression of early and late genes. We deleted the 3 out of the 4 immediate early genes, and therefore we obtained viral particles that are still capable of infecting the cells, but cannot replicate in the cells. And in this replication defective backbone, we inserted expression cassettes for FGF2 and BDNF under control of different promoters, and we can discuss the reason of that choice. We made some validation in vitro, and then we went in vivo to demonstrate that the vector can induce expression of these two transgenes. And again, to make a long story short, we obtained evidence of bilateral expression with a time course of expression of about 2 weeks, a little shorter for FGF2, a little longer for BDNF. This short time transgene expression is actually something desirable for this kind of approach, because a long-lasting or even a life-lasting overexpression of neurotrophic factors could be not detrimental for the function of the brain. So as a result of that, we ended up with a bunch of cells infected, and so we generated a number of cells that are now capable of expressing and secreting FGF2 and BDNF, changing the microenvironment of the tissue. We then moved to an epilepsy model, and specifically to the pilocarpine model. What we did was to inject pilocarpine and then stop status after 3 hours by injection of diazepam. And then we left the animals alone for 4 days, and only 4 days after status epilepticus, when the tissue is already damaged, and in particular the hippocampus is already damaged, only 4 days later we injected our vector. And then we follow the situation with two checkpoints. Checkpoint 1 is 14 days after status epilepticus, and it is about the time when animals start having spontaneous seizures in our hands. And at this time point, we checked for ongoing neurogenesis, for damage. And then we had another second checkpoint 1 month after status epilepticus, when all control untreated animals were having spontaneous seizures. And here we checked for the pathology and for the general outcome in terms of presence of spontaneous seizures. So this is checkpoint 1, and what you are seeing is the ongoing damage 2 weeks after status by labeling with fluorojade C. Essentially, what we see is that we can obtain an attenuation of damage, at least ipsilateral, to the injection, but this is not significant. The graphs that I will present from now on are all similar to this one, so let me tell you what it is about. I stands for ipsilateral to injection, the site where we injected the virus. C is contralateral. Yellow are naive animals. Gray are pilocarpine animals that were not treated with a vector or were treated with a control vector, and green are the animals treated with the vector expressed in FGF2 and BDNF. The situation is dramatically different in terms of neurogenesis. We monitor neurogenesis by multiple means, by using BRDU labeling, a marker of early differentiation, like nesting, and the pulse chase double labeling with BRDU and nesting. We also used other markers like SOX2 and so forth, and in the situations, and those gave consistent results. So what you see here is that status epilepticus, or if you wish, epileptogenesis per se induces an increase in neurogenesis, which is well known, and the treatment with the vector causes a further increase in neurogenesis. We asked how it was in terms of those aberrant aspects of neurogenesis that have been discussed in these days, like presence of ectopic cells in the heros and alterations in dendritic arborizations, like horizontal elongations in the new cells, and this was done using double coating. And as you see, this is an untreated animal, and all these aberrant aspects of neurogenesis are present. They are clearly attenuated in treated animals, even if neurogenesis is highly increased. Lastly, we wanted to see if at least a subset of these cells were entering the neuronal lineage of differentiation. So again, we did a pulse chase double labeling with BRDU and a marker of neurons. This pointer is not working out anymore, which is MAP2. And whereas untreated animals do not present any neurons, animals treated with a vector do have a large increase in the number of putative neurons, or BRDU labeled neurons. So this is checkpoint number 2. We are now 1 month after status epilepticus. And what we are looking at now is the pathological situation in the tissue. And this is triple labeling, or double as a matter of fact, because neurons, nuclei are labeled in blue with DAPI. Neurons are labeled in green with MAP2, and astrocytes in red with GFAP. This is a normal situation of a naive animal. I don't know. Okay. This is naive animal, and there is an obvious prevalence of neurons. This is an epileptic animal, and as you can see, there's an obvious loss of neurons in CA1 and in the hilus, and a massive astrocytosis. And this is an example of one animal treated with our vector, and as you can see, the situation is improved. I don't want to say it is perfect, but it is much better than under untreated conditions. So we also measured all the number of cells in different layers, and we observed a recovery of cells in the pyramidal gyrus and also in the hilus and in the oriens of CA1, which may indicate recovery of pyramidal neurons, so principal cells, but also of interneurons, and in particular, it was important to establish if there is a restoration of a balance between excitation and inhibition. So we measured the numbers of different kinds of interneurons, and we found in particular interesting a full recovery in the number of somatostatin interneurons. Somatostatin, as you know, somatostatin interneurons are involved in presynaptic inhibition. So this allows a speculation that the treatment may allow a preservation of cells, of neurons, in particular of interneurons, and this could produce a better control of excitability. Does that translate into a reduction of spontaneous seizures? The answer is yes. We performed 24/7 video EEG recordings in these animals, and we observed a dramatic reduction in the frequency of spontaneous seizures and also a reduction in their severity. So these are promising results, we believe. We need now to work on this concept and elaborate it further. But I would like to show you also some results that were obtained in an attempt to produce anti-seizure effects by other groups. And from this point of view, the first approach that has been used is to attempt to increase inhibition and to decrease excitation. The first, one example of the first approach is work done by the group of Amy Brooks-Cahal on GABA-A receptor subunits. It's been observed that in the pilocarpine model, there is a variation in the composition of GABA-A receptors in the dentate gyrus with a relatively down regulation of alpha or the alpha-1 subunit and an upregulation of the alpha-4 subunit. And this change is thought to implicate a lower activity of the receptor, a decrease in GABA currents, inhibitory currents. So Amy reasoned that if she could alter this situation by increasing expression of the alpha-1 subunit, that could be an approach to control seizures. And what she did was to generate an adeno-associated vector that was expressing the alpha-1 subunit under control of the alpha-4 promoter. She injected that vector in the hippocampus before pilocarpine. And then with pilocarpine, what happened was that the increase of the endogenous alpha-4 subunit was accompanied by a parallel increase in the alpha-1 subunit, and this decreased the frequency of spontaneous seizures. The other approach to reduce excitation has been taken by the group of McCall. And what it did was to generate adeno-associated vectors expressing in antisense subunits, in particular the NR1 subunit of the NMDA receptor. In antisense, therefore, reducing expression of the subunit. And this is very interesting because what they did was to insert in this gene in antisense under control of two different promoters. One promoter that is expressing principal excitatory cells, and the other promoter that is selectively expressed in inhibitory cells. And this is important because what happened was that when they were blocking NMDA currents in inhibitory neurons, they were having an increase in seizure susceptibility, whereas by inhibiting NMDA currents in excitatory neurons, they were having protection from seizures. And this leads to the concept, I have to say that more recently studies like this have been also performed by Matt Walker and Dimitri Kurman using the potassium channel and the lentivirus. But the concept is that when the transgene codes for a receptor or a channel, then it is very important to identify clearly the cell population where the transgene is produced, because it could be that you have some kind of effects of opposite kind of effects. One way to get around this problem could be to introduce, instead of receptors or channels, some soluble factors that will then activate the endogenous receptors that are at the site of injection. And this is pretty much what we did with neurotrophic factors, and it has been done, for example, by Merab-Kokaya using GDNF. But the most compelling evidence was collected with using the neuropeptides, the inhibitory neuropeptides, galanin and MPY. Galanin is a neuropeptide. There is strong evidence of implication, protective implication in epilepsy, both pharmacological and genetic. And again, the group of Macau developed a vector in which galanin was constitutively expressed because the gene was introduced in frame with the secretory signal of fibronectin, which is a constitutively secreted protein, and this way they obtained a constitutive secretion of galanin and a protection against the seizures, both in normal animals and in chronic models. And the evidence is even more compelling with MPY. Again, MPY is an inhibitory neuropeptide. I must say that MPY is a little more complicated because there are multiple receptor subtypes, and we know for certain that the Y2 and the Y5 subtypes mediate inhibitory effects, while the Y1 subtype is more uncertain. And again, by injecting adeno-associated vectors expressing MPY, it was possible to produce anti-seizure effects in multiple animal models. And I think it's particularly interesting what has been done. There are two interesting things here. The first one is what has been done by the group of Merab-Kokaya in Sweden, who induced overexpression of MPY together with its protective receptor Y2. And they obtained a potentiation of the effect. The other important thing is that what has been done instead by the group of Asla Pitkan and Anna Maria Vezzani, who injected the vector in chronically epileptic animals that were having spontaneous seizures, obtaining a dramatic reduction in the frequency of spontaneous seizures, actually, in the percentage of animals. A subset of animals had a reduction in the frequency of seizures, and there was also an attenuation of a progressive increase in seizure frequency. On top of that, other studies have shown that the vector was not producing significant alterations in other parameters like learning and memory, anxiety, and locomotor activity. So, to conclude, I think that gene therapy offers a number of opportunities for epileptology. We have the possibility of designing our vectors according to our needs in terms of spread from the site of injection, duration of transgene expression, and targeting specific cell populations. I also believe that epilepsy can be an opportunity for gene therapy because, at least for what refers to drug-resistant patients that undergo surgery after implantation of depth electrodes. It would be feasible ethically to inject vectors in the epileptogenic area of these patients during the first step of surgery. And the system would have a built-in rescue procedure because should the therapy not work, the patient would simply undergo surgery as originally planned, and the surgery would take away the epileptogenic tissue together with the vector. There's a lot that still remains to be done to get to this point, but promising results that have been obtained in other neurological diseases like Parkinson are encouraging, and so I believe that we will get to the point of having some clinical studies of gene therapy for epilepsy as well in the future. With that, I stop, and I don't know if I stayed in time, and thank you for your attention.